Hello everyone, welcome to the St. Joseph's open web-based learning platform, that is how you learn. Also welcome to Department of Biotechnology, St. Joseph's College for Women, Autonomous, Visakhapatnam. The Department of Biotechnology presents the SMOOC, that is St. Joseph's massive open online course in plant and animal biotechnology. It's a free online modular 8-week course for life science background students. Will be offered at the UG level.
Dear students, welcome back to Plant and Animal Biotechnology course. This is a module 4, Transgenic Animals and Gene Therapy. The chapters including production of healthcare products, in vitro fertilization, concept of gene therapy, transgenic animals. Taught by the Dr. A. Veerabrahmam, HOD, Department of Biotechnology, St. Joseph College for Women, Autonomous, Visakhapatnam. So these are the learning outcomes for the students. They acquire knowledge in production protocols of the healthcare products. They can be able to design and execute the protocols for in vitro fertilization. They can get an insight on a gene therapy, either ex vivo or in vivo gene therapy. So exact transferring of the corrected gene to nourish the damaged portion of the area or body, and able to be aware of merits and demerits in transgenic animals. So these are the contents for the present chapter, production of healthcare products, including insulin production, somatostatin, hepatitis B vaccine production, in vitro fertilization, gene therapy, transgenic animals. The first chapter, that is production of healthcare products, recombinant DNA technology that plays a very crucial role in production of insulin, somatostatin and vaccines. This technology generally allows the scientists to manipulate genes and insert them into host cells like bacteria or yeast, either E. coli or Saccharomyces cerevisiae, to produce desired proteins. So that is economic importance, commercial basis. So that from biotechnology, recombinant DNA technology is a promising area to produce the healthcare products. And these are the steps in recombinant DNA technology. First of all, you need to isolate the genetic material, cut the genetic material, join the DNA with the required vector molecule, insert the recombinant vector into the host cell, desired host cell, and select the host cell and screen them for the production of the desired gene inserted. And whatever the cells isolated or say screened are subjected to the isolation of the expressed or perpetuated molecule, and with the help of the desired promoters, they will be expressed to get the protein product. So isolating the gene of interest, for example insulin, somatostatin, or a pathogen antigen or a vaccine producing gene is isolated from its source. Restriction enzymes are the best choice to cut the DNA at a specific sites, and desired gene is then joined with a plasmid vector. There are a lot of vectors available, plasmids, plasmids, phasmids, or yeast artificial vectors or shuttle vectors, right? And engineered vectors also available to perpetuate the molecules. Inserting into the host cell, the recombinant plasmid is then introduced into the host cell like E. coli or yeast. Cells that have successfully taken up the recombinant plasmid are selected and screened. The host cell expresses the gene of interest, and the protein product is harvested and purified, then packaged and marketized based on the requirement in the society. So it will be prepared. So first of all, let us discuss the production of insulin. Recombinant insulin, that is produced by inserting the human insulin gene into bacterial plasmids. PBR322 is the best choice, which are then introduced into the bacteria like E. coli. These bacteria then produce the insulin, which is harvested and purified. Eli Lilly company is for the first time they produced the recombinant insulin in the name of inulin. The insulin produced by E. coli can be further processed to create different types of insulin, including recombinant long-acting insulin analogs. So analogs are very best choice for the diabetic patients. So biofarming is the where insulin genes are inserted into plants. So of course, there's a present hot topic also being explored to reduce the production costs. So this is the protocol or the schematic representation for the uh, production of human insulin on the name of humulin yum. So this is the recombinant molecule. First, we need to prepare a vector molecule with the help of the restriction endonucleases. A promoter must be included, an antibiotic, a marker gene, and another marker gene, beta galactosidase gene. So once any foreign DNA or a gene is inserted in any of these two, their function will be lost. That is known as insertional inactivation. So that every time to identify whether the recombinant cells are there, it must not be damaged. So insulin gene is as a subunit inserted into the vector molecule, most probably a PBR322, right? And insert the recombinant vector into a E. coli. So where the E. coli promoter can recognizes in addition to their normal genome. So the beta galactosidase gene will be expressed and forms the culture only transformed cells. Only transformed or a recombinant cells can produce this the insulin mRNA, and that mRNA will be translated into the protein like this hormone, insulin hormone. And first of all, chemically remove insulin polypeptides from the beta galactosidase protein, and then each A and B chain such a way prepared in the E. coli cells are joined with the disulfide bond, then it will be active insulin. Clear? So this active insulin combine the polypeptide to produce functional insulin. Clear? So then it will be for the commercial uses or pharmaceutical uses for the diabetic patients. The next topic, production of somatostatin, is a hormone that regulates growth, can also be produced using recombinant DNA technology. Genetically modified organisms can be used to express the somatostatin gene, and the somatostatin can be harvested and purified, marketized, commercialized. Fermentation process are used to cultivate the cells expressing the somatostatin gene, and the somatostatin hormone gene can be separated from the fermentation broth. This is a schematic representation of how the somatostatin can express or produced from the recombinant DNA technology. From the human cell growth hormone, a gene for human growth hormone is isolated and which is cleaved with the EcoR1 restriction endonuclease that develops the staggered ends, okay, or sticky ends. EcoR1, which is a restriction endonuclease, they specifically cuts the DNA sequence G and A, guanine and adenine. Between the two disulfide bridges, phosphodiester bond will be cleaved, and then the same enzyme will be utilized for the preparing the bacteria. So choose a bacterial cell, either a EcoK12 is the best choice, a bacterial chromosome, and plasmid is isolated from the bacteria, and EcoR1 cuts the sequence, plasmid sequence, prepares the sticky ends, where already the sticky ends are ready in case of the growth hormone gene, and inserted through the DNA ligase, and polymerase seals the gaps and it is a now human gene for human growth hormone recombinant DNA, and this recombinant vector molecule is inserted into the bacterial cell, that is E. coli. Now the E. coli cells are inoculated onto the or suspended into the suspension culture or a fermenter, in a fermenter, seeded in a fermenter. So based on the requirement, the bacterial cells are cultured for the growth hormone. Then the cells are harvested again. Whatever recombinant cells, the screened variety of techniques, variety of reagents are isolated. Then it is purified through desired protocols, sustained protocols, to get the somatostatin hormone. Next topic is production of vaccine in this chapter. Hepatitis B. Hepatitis B, which can which can control the hepatitis B virus, jaundice, are produced using recombinant DNA technology by inserting the gene for hepatitis B surface antigen, not entire hepatitis B virus, into the yeast cells. Here, yeast cells are the host cells where it accommodates the long lengths of the surface antigen gene, which then produce the antigen. The purified HBs antigen is then used to create the vaccine, stimulating the immune system without causing infection. Okay, so let us see the steps involved in the production of vaccine, that is hepatitis B: gene insertion, yeast production, purification, vaccine formulation. The gene encoding HBsAg, surface antigen, hepatitis B surface antigen, which is the protein on the surface of a hepatitis B virus, isolated, inserted into the host cells, that is yeast, Saccharomyces cerevisiae, Bacchus yeast, right? The yeast cells now harboring the gene are cultured and they begin to produce surface antigen as a protein. Purification. HBs antigen protein is then harvested and purified from the yeast cells. This process involves breaking down the yeast cells and using chromatography to separate the HBs antigen from other cellular components. Okay, so either of the exclusion or size exclusion chromatography used to separate the surface antigen, since it is a protein, so that from other cellular components the protein will be isolated. Vaccine formulation. So this is the very important step during the vaccine production. The purified surface antigen is then formulated into vaccine. This often involves combining it with an adjuvant. Adjuvant is a molecule, a substance that enhances the immune response, such as aluminum hydroxide. So by its own, they may not enhance the immune response. Once it is combined with the any protein which may be comprising 10 kilodalton size potential antigen, of course sometimes it may not enhance the illicit immune response in the body, then adjuvants can be used. This is the schematic protocol for the hepatitis B production. The next topic in the chapter is IVF, in vitro fertilization process. Is a fertility treatment where a woman's egg is combined with a sperm in the laboratory dish outside the body to create an embryo. This process, also known as assisted reproductive technology, ART, is used to help women who have difficulty conceiving naturally. Let us see how IVF works. Ovarian stimulation. Hormonal medications are used to stimulate the ovaries to produce multiple eggs instead of just one. So here, multiple egg production in the women where the difficulty in conceiving, of course a painful process, of course more nutrition is required to secrete or to produce the more eggs, ovaries. Egg retrieval. Egg retrieval. The eggs are retrieved from the ovaries under anesthesia. Of course, this is also a painful process. Next step is sperm collection. The male partner's sperm is collected, or donor sperm may be used to fertilize the egg. The next step is fertilization. The eggs are combined with the sperm in a laboratory under aseptic conditions, and fertilization occurs. The process is known as in vitro fertilization. Within the glass or within the laboratory, the process is known in vitro fertilization. No involvement of the physical body contact. Embryo development. The fertilized egg is now an embryo, is allowed to develop in the lab for a few days in a specialized environment, a specially designed buffering solution that nourishes the outer surfaces of the egg or an embryo. Embryo transfer. One or more embryos are transferred into the woman's uterus, so where they possibly nourished to get the embryo to fetus to organism or different stages, 256 cell stage, blastula, blastopore, and then pregnancy. If the embryo implants in the uterine lining, pregnancy occurs, so that the hormone, that is secondary sexual hormone, will be released. Infertility. The reason. What is the reason for IVF? Infertility, inability to conceive naturally, genetic problems that may be passed down, and problems with the ovulation or a sperm quality. No particular sperm or a sperm had. So there are some risks associated with the IVF and success rates, important considerations. So let us see what are the risks associated with the IVF. Multiple pregnancy. Since once you isolate the number of embryos or eggs, so at a time, if they are flooded with the male pronucleus, more than one baby will be there. So in such cases, miscarriages will be happen. And another is ovarian hyperstimulation syndrome. So this is a way, of course, 1 in 10 cases will be occurred. Infection. So every time this is a surgical operation, need to be implanted the embryonated egg into the uterus. So bleeding will be there, damage to the bowels or bladder. So this is a risk factor in case of IVF. IVF success rates depends on several factors, including age, the cause of infertility, the quality of eggs and sperm. Okay, and age is the one constraint to success the IVF. IVF is a complex and expensive process. So most probably 1 lakh to 5 lakhs can be charged for a sitting. So that the couples need to think about the once they are going to or optimize to get the IVF process. It may be involve multiple cycles and emotional and physical strain. Important to have realistic expectations about success rates. IVF may involve the use of donor eggs or sperm or gestational surrogacy. So this is the protocol schematic representation of the in vitro fertilization. The next topic in this chapter, concept of gene therapy. Gene therapy is a medical approach that aims to treat or prevent diseases by directly modifying a person's genes. It involves the introducing a functional gene into correct a defective gene, or introducing a new gene to help or fight a disease. This technique offers the potential to treat conditions at the root level by targeting the genetic cause within cells, unlike traditional drugs that often address symptoms. So important goals for the gene therapy, to replace the faulty gene with a healthy copy, to inactivate a malfunctioning gene, to introduce a new gene to help or treat a disease. Let us see how it works. Introducing new genetic material, either DNA or RNA, into cells, often using vectors like modified viruses, because The natural viruses may create a problem or viral diseases, viral replications that do occur irrespective of the object carried out by the virus. Somatic gene therapy, modifying cells in the body like stem cells to treat a single person's condition. Germline gene therapy, modifying cells that can be passed down to future generations, not currently approved because it is a more ethical issue in the gene therapy system. So there are two approaches available in gene therapy, in vivo and ex vivo. In vivo, directly delivering the gene into the target cells in the body. In ex vivo, removing the cells from the body, modifying them in the laboratory, that is in vitro, outside the body, preparing them for or adapting to the conditions and then returning them to the patient. So genetic diseases, cancer, infectious diseases are the applications of this concept of the gene therapy. Cystic fibrosis, muscular dystrophy, Huntington's diseases are effectively controlled by the gene therapy. And then cancer, gene therapy can target cancer cells or enhance the immune system's ability to fight cancer. Some diseases, some studies are exploring gene therapy for treating HIV and other viral infections. Of course, not yet proved, but there is a hope to control the viral load in the HIV patients. Until unless the viral load is identified as a lethal condition. So safety, delivery, targeting the gene into the body is very, very important key considerations. Is still relatively new, and there is a ongoing research about potential side effects such as immune responses, unintended mutations. Delivery efficiently and safely delivering the therapeutic gene to the target cells is a major challenge because so many obstacles are there. During the transportation, the gene must not be modified, must not be mutated, must not be deviated, or not to be dormant, so it must be active. Precisely targeting the right cells, avoiding unwanted effect is crucial in the gene therapy process. So this is the schematic representation, diagrammatic representation of the gene therapy, ex vivo and in vivo gene therapy. Next chapter, next topic in the chapter, concept of transgenic animals. Transgenic animals are animals whose genomes have been altered by insertion of a foreign gene, that is transgenic, from another species, typically through genetic engineering techniques. That gene can be introduced into the one animal or one organism. This process allows for the introduction of specific traits or the study of gene function and expression in a controlled manner. So DNA modification, foreign gene expression are the key characteristics. Transgenic animals have their own DNA modified by adding or altering genes, creating a genetically modified genetically modified organisms, that is gene modified organisms. Foreign gene expression, the introduced foreign gene is expressed, meaning the animal produces a protein coded by a gene. So these are the methods for the transgenic animal production: microinjection, embryonic stem cell mediated transfer, sperm mediated transfer. So these are the most available methods for the transgenic animal production. Directly injecting foreign DNA into the pronucleus of the fertilized egg, introducing DNA into embryonic stem cells, which are then used to create transgenic animals, that is the second approach. Incubating sperm with foreign DNA and fertilizing eggs in vitro or in vivo, that is the third approach. So research, therapeutic protein production, vaccine safety testing, agriculture improvement are the main applications for these transgenic animals. Studying gene function, development and disease models. Producing human proteins like insulin or growth factors for the diabetic patients or growth less persons. Developing models for testing vaccine safety before human trials. Improving traits like milk production or disease resistance in livestock. The introduced transgene is passed on to the offspring, making the trait heritable. Transgenic mice, transgenic sheep, transgenic chicken are as of now prepared, commonly used model organism to research due to their relatively simple genomes and high reproductive rate. Transgenic sheep or a goat produce proteins in their milk. Transgenic chicken used to produce proteins in their egg whites. So these are the important concept of the genetic animals. Milk protein for the goat and sheep, and regulatory reason, coding reason, foreign gene are added. So in that way, transgene is prepared and produced into the transport into the organisms. So these are the references for the topics discussed as of now. Thank you for your attention. Thank you very much.